Good morning, grade 12 students. In this session, we are going to review chapter 9 about the function of neurons. We're going to make an overall revision for chapter 9 for the different documents. Let's start with document 1 about the resting potential on your book on page 182. And the resting potential, we determine that we have a difference in the potential difference. We have differences in the delta V between the interior and the exterior of the nerve fiber. So we have a difference of about minus 70 millivolt. The interior is more polarized than the exterior. It's polarized, meaning that it's negatively charged, and it's the resting potential. It's equal to minus 70 millivolt. Here I'm talking about human. It differs according to the species. Origin of the resting potential, we said that the origin is the difference in the concentration of the ions between the intracellular and the extracellular. And you have to know that we have selectivity and the membrane like we did. So we have selectivity in the membrane. This allow the movement of the ions in opposite direction. So they allow the movement of Na plus and K plus ions in opposite direction according to the gradient. So here you have to know that the origin is mostly the difference in the ionic concentration due to the difference in the permeability of the membrane potential. Second, we talk about the source of energy inside the cell, which is called the ATP, adenosine triphosphate. This adenosine triphosphate is the source of energy, and it allows the diffusion or the movement of the ions through what? Through a special pump. It's called the Na plus K plus pump. This is called an active transport where the energy is needed. Energy is needed for the transportation of the ions into inside or outside. So K plus they enter and Na plus they move outside through the Na plus K plus pump, and they need energy. This energy is in the form of ATP, adenosine T tri P phosphate. So adenosine triphosphate. This pump is called the Na plus K plus pump, or they call it Na plus K plus ATPase. ATPase meaning that it uses the ATP in the form of energy to allow the fusion of the ions. So here we have a potential difference that exists between the anterior and the exterior of the nerve fiber. This potential difference is equal to minus 70 millivolt between the anterior and the exterior in human. This means that we have a polarity, meaning that we have negativity, and this negativity is equal to minus 70 millivolt. Concentration in the ions in the intracellular and the extracellular, they differ, and this is the origin of the resting potential. So unequal distribution of the ions on both sides of the membrane, meaning that the internal and the external side of the membrane. And we have selective permeability, meaning that they allow outflow of K plus more than the inflow of Na plus. So بيطلع Na plus أكثر من K plus أكثر من Na plus راح يفوتوا. So this will lead to the appearance of the resting potential, and this resting potential is equal to minus 70 millivolt. Here about ATP ADP adenosine triphosphate ADP adenosine diphosphate. So DMP اتفقنا هو داينيتروفينول. داينيتروفينول it inhibits the phosphorylation، يعني بيمنع يتحول ADP into ATP، بيمنع نزيد له phosphate group. So here we have differences. So this will lead to the flow of Na plus needs more energy in the form of ATP. The outflow of Na plus ions need more energy. This energy should be supplied in the form of ATP. Also, talking about the pump, Na plus K plus pump permits the pumping of what? Na plus and K plus against the concentration. So Na plus concentration in the extracellular is more than that in the intracellular, as well as the concentration of K plus in the intracellular is more than that in the extracellular. So this will lead to inflow of Na plus and outflow of the K plus with the concentration gradient. This will lead to the appearance of the resting potential minus 70 millivolt, and this will stay at minus 70 unless we have an effective stimulation. Also, we said about diffusion. What is diffusion? Is the flow of a substance from a medium of high concentration to a medium of low concentration with gradient. Passive and active transport. What are the difference? We said that the active it needs energy, while the passive, it doesn't need energy. So active transport, like the case of Na plus K plus bond, it needs energy in the form of ATP, which is the source of energy used by the cell. ATP, adenosine triphosphate. So ADP upon phosphorylation, what the meaning of phosphorylation? Adding a phosphate group. So ADP upon phosphorylation will lead to ATP. This is the source of energy. While dephosphorylation, what the meaning of dephosphorylation? Removal of a phosphate group will lead to the appearance of ADP again. Moving now to the action potential. We recorded the different phases of the action potential on the oscilloscope using the stimulating electrode, recording electrode, and the reference one, which is placed inside the seawater. So here we have different phases of the action potential. We have at minus 70 millivolt, which is number one on the graph of document B. So number one is called the resting potential, and the delta V, the potential difference, it's equal to minus 70 millivolt. Then upon stimulation, we have a hypopolarization. This hypopolarization will lead to the increase in the potential difference. Maybe it will lead to the increase that reach the threshold of depolarization and will fire an action potential, or it's maybe a small hyperpolarization artifact that will not exceed the threshold of depolarization. Three, we have the depolarization. We have moving away from the negative toward the positive. Phase number four is the repolarization is coming back to the negativity. And five is hyperpolarization, negativity more than the usual. And six, returning back to the resting state, minus 70 millivolt. So here we said about the difference in the phases. These difference in the phases, they depend on what? They depend on the number of channels. They depend on the number of Na plus channels and the number of K plus channels according to the different phases. What will happen through these phases? So here we said that during depolarization, the number of Na plus open, it increases. So the number of K plus is almost null. So the potential difference, it increases from 70 to 30, minus 70 for sure, to 30 millivolt. So here, depolarization is caused by what? You have to know this. Depolarization is caused by the opening of the sodium ions channels, of the Na plus channels, and this will lead to the entrance of Na plus from the extracellular toward the intracellular. This is the first important note. You have to know this, and you have to study this indication. That's why I put it in red. So depolarization is caused by what? The opening of Na plus channels. Na plus channels, the Na plus ions, they enter inside the cell, inside the intracellular, and this will lead to the depolarization, decrease in the negativity and movement toward the positivity. Also, repolarization, going back to the negative, just due to what? Due to the opening of the other channels that are the calcium channels, K plus channels, and this will lead to the flow of K plus from the intracellular toward the extracellular. So here, till now, we have two different phases. First one, depolarization, is because of the Na plus, it moved from the extra to the intracellular medium, and the K plus is for the repolarization, so the opening of these channels and the flow of K plus from the intra toward the extracellular medium. And also during hyperpolarization, what happened? Flow of some amount of K plus ions from the intracellular to the extracellular, and this will lead to the more negativity. So the action potential is caused by what? The change in the permeability of the membrane of the nerve fiber to both ions, Na plus and K plus ions that are interfering mainly in the phases. Now, let's move to document 3. Document 3 illustrating about the nerve impulse and the action potential. So here we are talking about effective stimulation, ineffective stimulation, threshold of excitation, and their differences. So as we see before, that the nerve fiber is obeys the law of all or none. Either we have an action potential or we don't have at all. This action potential is due to an effective stimulation. Intensity is I1, I2, I3, I4, whatever. So I2 and I1 and I2 and I3, they didn't cause an action potential since the stimulation is ineffective. and the potential difference or the membrane potential did not exceed the threshold of excitation. However, I4, I5, and I6 they showed an action potential since the membrane potential it exceeds the threshold of depolarization, and these action potential they have the same amplitude. And this is another conclusion about the action potential or the transmission of the nervous message in a nerve fiber. It obeys the law of all or none. Either we have an action potential or we don't have at all. These action potentials, all of them, they have the same amplitude. Nerve impulse inside the nerve, you have to know that we have different factors that affect the speed of propagation of the nervous message. We have the diameter. As diameter increases, the speed of propagation of the nervous message increases. We have also the myelination. As the myelination exists, the speed of propagation of the nervous message increases as well. Also, we have, other than the diameter, other than the myelination, we have the temperature. As the temperature increases, the speed of propagation of the nervous message will increase as well. So here we have the recording of potential in the nerve with increasing intensity in figure 1, document E. And we have different distances. So as the distance increases, we have two different action potentials, but they have different amplitudes. This is related to the distance as move away from the case of the site of the stimulation. So here also we continue to talk about the different parts of the nerve fiber, myelinated, non-myelinated, what happened during this. We said that the nerve fiber, the nerve is conducted with constant amplitude, as we said, the amplitude is constant, but the difference in something called the frequency. So myelination at present, so the nervous message speed will increase, and as well as for the diameter. So also we have other factor than the diameter and the myelination. We said that we have temperature. So as temperature increases, the speed of the nervous message or the speed of propagation of the nervous message will increase as well. And we said that we have different distances, and the distance increases between the two stimulations or the site of stimulation, the nervous message will be slower. So maybe it's due to the presence of myelinated and non-myelinated. Maybe we have large diameter, maybe you have small diameter. That's why we have different in the speed of the propagation of the nervous message. Moving for document 4, in document 4, we talk about the sensory receptors and the nerve impulse. We said about the different types of receptors. We have mechanoreceptors that respond to the stimulus, that is the touching, pressure, stretching, and so on. Mainly the sensory organs, they are the muscles, the vessels, and the skin. Also, we have thermoreceptors that detect the changes in temperature, like the skin and the tongue as well. Also, we have photoreceptors. So these photoreceptors, they respond to the nervous message in the eye mainly, and they respond to the light stimulus. And also we have the pain stimulation, which is called the nociceptors. And also we have the chemoreceptors that detect the changes in the level of the concentration of some ions, some chemicals, and these are the level of the tongue as well as the level of the nose. And also we talk about special type of receptors. They are called fascinocorpuscles. They are touch receptors. They are sensitive to pressure. So they are mechanoreceptors since they respond to the stimulus of pressure. So they are mechanoreceptors. Also, we have, according to their location, we have extra receptors and enteroreceptors. So the exteroreceptor, they receive information from the external medium, while the entero, they provide information or they receive information about the internal medium. So any light, sound, pressure, or something, this is related to mainly to the external. So exteroreceptor, they come from the external medium. Also, we talk about the relationship between the intensity of the stimulus, the receptor potential, and the action potential. So as the intensity of the stimulus increases, the potential difference as well as will increase, and this will lead to the change in the potential and the frequency of the action potential. So here we can do that frequency is the one that codes the action potential or the propagation of the nervous message. So this is the role here. So the nervous message inside the nerve fiber obeys the law of all or none. This is the first conclusion. Second, either we have an action potential means or we don't. So this is the law of all or none. Second conclusion is related to the frequency, that the nervous message is coded by frequency and not by amplitude. All these notes that I'm explaining, highlighting this, different colors, different, for example, fonts, all of them, they are very, very, very important. You can't use the same ideas and you don't have the same ideas in all the questions. But mainly, all the questions, they have the same conclusion or they have the same indication. So if you know that the nervous muscle is coded by frequency and not by amplitude, even though I changes the form of the question, I worded the question in different mode and gave you frequency and gave you amplitude. You know that the conclusion is going to be that the nervous muscle is coded by frequency and not by amplitude. Since the amplitude it remains constant, it remains the same. For example, so this is the indications or the conclusions. They are very, 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 very important in order to be ready for the question. So if you know the concept of the question and already you know or you study or you memorize the indication or the conclusion, so you can solve the question easily. Already you know the conclusion, already you know the indication, or already you have information about the conclusion and indication. That's it. So these conclusions and indications, they are very important to focus on and to work on. So here, conclusions and indications, they are very important to focus on because they will help you a lot to answer the questions, to be ready for any question, even though you have some problem in understanding the question, it's somehow complicated, but if you move to read the question and you read the figures, you know that it's talking about frequency, it's talking about the action potential, the intensity, about, for example, the amplitude, about the frequency. So they are comparing. So my conclusion is going to be that the nervous muscle is coded by frequency and not by the amplitude and so on. So these are very important. So you have to be smart. You have to be smart in uh, focusing on the question, knowing the question, what does the question need, شو بده السؤال, and then I have to focus on my conclusion and my indication. So these indication and conclusion that I highlight them in blue and red and different colors, this means that they are very important to help you to understand the question and the concept of the question. So there, for example, the nervous muscle and nerve fiber is coded by frequency and not by amplitude. You're going to use this a lot. Whenever you have a question talking about the frequency, I'm talking about the amplitude of the nervous muscle and the nerve fiber. So these questions, these indications and these conclusions, they are very important. They help you to focus on the question and to answer them. Moving to document 5, when we talk about the synapse, the structure and the function, we said we have different types of synapses. Either they are excitatory synapse that will lead finally to the excitatory postsynaptic potential, EPSP, or the inhibitory postsynaptic potential that leads to inhibition at the level of the postsynaptic terminal. We said that we have action potential. This action potential, it arrives toward the presynaptic terminal. Then we have fusion of CA2 plus. This CA2 plus allows the secretion or the exocytosis of the neurotransmitter inside the secretory vesicles, where into the synaptic cleft. This release into the synaptic cleft will lead to the binding on the receptors on the postsynaptic, and this will lead to the postsynaptic potential, PSP. This PSP is of two types, postsynaptic potential. Either it's I, it's inhibitory, or it's E, excitatory. So if it's inhibitory, we call it IPSP, inhibitory postsynaptic potential. While if it's excitatory, it will be EPSP, excitatory postsynaptic potential. So that depends on the type of the synapse. If we have propagation of the nervous muscles and the nervous muscle propagated from one synapse to another, from one neuron to another, from a neuron to a cell, from a neuron to a gland, from a neuron to a muscle. So this message is going to be propagated. So this synapse between them is going to be excitatory. While if the nervous muscle is inhibited or it's, for example, decrease in the value or the intensity or the amplitude, this means that this synapse is going to be an inhibitory 1. So these you have to know that the message is conducted from the pre to the post. All of you have to know this. The message is going to be from the pre to the post. Nervous message is not going to move in the opposite direction. That's why we said that it moves in a unidirectional manner from the presynaptic terminal toward the postsynaptic and not in the opposite. And you have to know that action potential at the end of the pre, it causes. Yes, Muhammad, you have a question before we continue?
Yes, it says drugs acquired للquiz.
Drugs, meaning that you are talking about
Antagonist.
agonist, only the drugs that we saw, we explained them during the introduction for the nervous system. These one are required, yes,
Okay.
because we are going to talk about them in the neurotransmitters in chapter 12. Neurotransmitters and the medical applications. We are going to talk about the acetylcholine, about the GABA and other neurotransmitters. But the one that I explained them in the introduction, yes, they are required. By the way, all the notes that are explained from the beginning of the nervous system during the introduction and during chapter 9, all of them, they are included in this course. And you don't have to focus only on chapter 9, even though chapter 9 includes most of the information, but you have to go back also to the introduction. So we said that the action potential that arrives at the level of the presynaptic terminal, it provokes or it leads or it facilitates or it helps the entrance of Ca2 plus of calcium ions into the presynaptic. So find the action potential to Ca2 plus and the presynaptic. This will lead to the transmission of the message to the post. How? Because they lead to the exocytosis of the neurotransmitters. One of them is here the acetylcholine. It's secreted by exocytosis of the neurotransmitter acetylcholine into the synaptic cleft, and this will lead to the propagation of the message from the pre to the post. So here you have to know that action potential reaches to the pre. This will lead to the entrance of Ca2 plus, Ca2 plus in the presynaptic terminal. This will lead to the exocytosis of the neurotransmitters, like this case, they are acetylcholine from the secretory vesicles inside the synaptic cleft, and this will lead to the conduction of the nervous message from the pre to the post synaptic terminal. So here, as you know, the amplitude of the potential in the postsynaptic is directly proportional to the amount of the acetylcholine. كل ما زدنا الامount of acetylcholine كل ما زادت the amplitude of the action potential and vice versa. So action potential, this is the conclusion. Reaching the end of the presynaptic causes the entrance of Ca2 plus. This Ca2 plus will lead to the exocytosis of the neurotransmitter inside the secretory vesicles. There is acetylcholine, ACH. This will cause the transmission of the message from the pre to the post, where the amplitude is directly proportional to the amount of acetylcholine. And you have to know the acetylcholine and curare, they are antagonistic. One of them causes excitation and the other one causes relaxation of the muscle, as we said before. So differences between voltage dependent and the chemically dependent. Voltage dependent channels, they depend on the voltage. Difference in the membrane potential. However, chemically dependent channels, they open and close due to the binding of chemical substances to receptors found in the channels. Document 6 talking about the integrating properties of the nerve centers. Already we said about the excitatory and the inhibitory synapses, about IPSP, excitatory postsynaptic potential. We don't have an action potential even though it means as the resting or it means as a negative. However, EPSP, excitatory postsynaptic potential, they will lead to the increase in the level or the amplitude of the action potential. As we said before, that we have something called spatial summation and we have something called temporal summation. Spatial, two nervous muscles coming from two different synapses or more. By temporal summation is the summation of the nervous muscles coming from where? From the same synapse. For the summation to take place, either the temporal or the spatial, it should be separated by a short period of time. So here, the message that are subjected to summation, they should be separated by a small period of time. So these, everything related to the notes from chapter 9, document 1 till document 6. So these six documents, we covered them. In the next session, we're going to continue solving the exercises of the chapter. Next week, we're going to start with chapter 12, and then we can move chapter 12, solving the exercises of chapter 12, and then we're going to move to the immunology part.